So hi everyone, my name is Ina Ricardo-Lax. I'm a 6-year postdoc at the lab of Charlie Rice at the Rockefeller University. And today I'm going to tell you about our SARS-CoV-2 replicon system that we built. So SARS-CoV-2, the cause for this COVID-19 pandemic, is a biosafety level 3 pathogen, which means that we need special facilities and equipment to work with the infectious virus. And here in this photo, you can see me in full gear. And in the background is our BSL-3 facility, which we're really lucky to have. But in fact, most labs don't have access to biosafety level 3 facilities. And even those that do, most of the BSL-3 facilities are not equipped for certain kinds of experiments, such as high throughput experiments. And so this really makes a slow progress studying SARS-CoV-2 and testing different drugs. So we decided when the pandemic hit, we decided to make SARS-CoV-2 replicons. And what are viral replicons? Viral replicons are self-replicating, in this case, RNA molecules that when you put them in the cells, they encode for all the genes that they need to replicate the RNA, but they lack either some or all of the structural genes that allow them to make infectious virus. And so these replicons, when you put them in the cells, they can get you sustained replication that you can study, but no infectious viruses coming out. And this has a very long history of use at biosafety level 2. Another advantage of replicon system is that you can engineer them to express reporters, which makes it easier to study and quantify. And then lastly, these replicons are a very efficient drug screening method. So for example, hepatitis C virus replicons that were developed in our lab previously in other labs were used to find hepatitis C virus antivirals that today are used very successfully in the clinic. So with SARS-CoV-2 replicons, there are some challenges. First of all, that the RNA genome is very long, it's about 30 kb. And the other trouble is that some of the coronavirus sequences are actually toxic in bacteria. So this makes it really problematic using just standard cloning techniques, and we have to use something else. And in addition, whenever you want to use a viral clone that you will transfect in the cells and will make infectious virus, you only need a little bit of genetic material. It's enough to have a few RNA molecules that will start the process. But with replicons, for each and every experiment, you need large amounts of high quality RNA. And so to try answer these first two challenges, we collaborated with the lab of Volker Thiel in the University of Bern in Switzerland. And they had a system where you can make overlapping DNA sequences that when you transform into yeast, because the yeast have a very efficient homologous recombination mechanisms, they will actually assemble the whole genome for you in a very high efficiency. So we assembled our replicon, and this is the design of our replicon. You see these overlapping DNA fragments. And what I want you to notice mostly is that this gene is the spike gene. And we completely replaced the entire coding sequence of spike with some selectable reporters. And we have a nuclear localized neon green reporter and a secreted Gaussian luciferase, which I will use interchangeably throughout my talk. So when you take this replicon and you take DNA, you transcribe in vitro the RNA and you electroporate this RNA into cells, you can get to a pretty good efficiency in launching the replicons. You can get up to 20% of cells that will give you reporter readout. So what can you do with these replicons? First of all, you can test drugs. This is just an example. We used it to test remdesivir, which is a known inhibitor of the viral RNA polymerase. And it will remdesivir will inhibit the replicon in very similar concentrations as we see with the actual virus. As a negative control here, we use an inhibitor of entry. And because by using replicons, we really bypass the entry because we're just putting in directly the RNA into cells, we don't expect entry inhibitors to work on replicons, and indeed we don't. Next, we wanted to see whether our replicons can be used to study virus-host interactions. And in this case, what we used was TMM41B. This is a host gene that were found in our lab last year by using CRISPR screen. And we found that it's very important for virus infection. And now when we use knockout cells for TMM41B, we see that the replicon is inhibited in these cells. But if you reconstitute TMM41B, we get a partial rescue of the reporter signal. And so lastly, we wanted to see whether SARS-CoV-2 replicons can be used to study viral variants. In this case, we used NSP1 mutant. So NSP1 is a protein that is not strictly required for viral replication, but what it will do, it will bind to the ribosome and it will prevent the host protein expression. The reason why it does that is to block activation of antiviral program in the infected cells. And so what we did, we mutated the NSP1 in such a way that it can no longer bind the ribosome. And what happens in the replicon, so what we see here is that the replicon signal is not actually very different because, as I told you, it's not strictly required for viral replication. However, if you look at cell viability, the wild-type replicon is cytotoxic to the cells because it will prevent their protein expression. But this mutant, the cells are happy and viable. So because NSP1 is needed for antiviral program of the cells, what will happen if we now activate this program by treating the cells with interferon? So when we treat cells with interferon, the NSP1 mutant replicons are very sensitive to interferons because they cannot block this antiviral state of the cells. But the wild-type replicon is much less sensitive. So just to summarize this part, I've shown you that we were able to assemble our replicons in yeast, and we were able to get efficient RNA launch in cells. And these replicons, I've shown you that they can be used for drug testing, for looking at virus-host interaction, and also characterizing viral variants. So this is all great, but we have still some limitations because the replicon launch is highly artificial. So first of all, we need to electroporate cells. And even if we do use something that's not as harsh as electroporation, we're still putting a lot of foreign RNA molecules into cells. And really not a lot of cells can tolerate it in the aspect of RNA sensitive toxicity. And this really limits the type of cells that we can use. So to overcome this challenge, we decided to build SARS-CoV-2 replicon delivery particles, or RDPs. And those are essentially virions, but they can only infect once. So they're single cycle virions. The way it works is that we transfect cells with spike. And these cells now express spike, and we electroporate them with replicon RNA. This replicon RNA does not encode for spike. So only these producer cells that have spike will be able to package the replicon and make infectious particles, which will infect once. But these recipient cells, because they don't express spike and the replicon doesn't express spike, this, if we take the supernatant from these cells and putting on new cells, that is supposed to be clean. And this was a key consideration in our work to make sure that these variants are indeed single cycle and safe to use. So this is just showing you how it looks. Both colors actually reflect the replicon here. And you see these are the producer cells electroporated with the replicon, and they show nice signal. But only if you have spike expressed in these cells, then you will have RDPs. You can infect the next batch of cells. But if you take supernatant from these cells, you put it on naive cells, you see nothing. So this means that they're really single cycle and clean. So now we essentially have SARS-CoV-2 virus that is only single cycle infectious, and we can actually use that to look at entry steps. And in this case, we used two different spikes and tested them against two previously characterized monoclonal antibodies, neutralizing antibodies. And what I want you to appreciate from this figure mostly is how similar these neutralization curves look between the RDPs and the actual infectious virus. So this really means that we can reliably use our RDPs in neutralization assay to test different spikes and different neutralizing antibodies. So this is great, but this is still a little limited because we need cells that overexpress ACE and TMPRSS to get it to work. So we then decided to take a page out of the lentivirus book. And if you ever worked with a lentivirus, you know that you usually use the vesicular stomatitis virus glycoprotein, or VSVG, to coat your virus particles. And the advantage of using VSVG is it can target most of the cell types that you know of in many, many hosts as well. So we did the same thing for SARS-CoV-2. So basically, instead of expressing spike, we expressed VSVG. And we made those VSVG RDPs. And indeed, these can basically target all the cell types that we've tested. For example, these A549 cells, they don't normally express ACE2, so the receptor for SARS-CoV-2, but we were able to use those. And these two are primary cells, and we can later use them to show that they can be used for testing different drugs in them. So this is, for example, with remdesivir and interferon alpha. So to summarize the second part, I've shown you that RDPs can give you authentic one-way infection. It solves the harsh delivery problem. It's an isogenic platform, so you can test different spikes without having to re-engineer with the replicon. And we can use Vsvg to target different cell types. So I want to thank you for listening. I want to thank my mentor, Charlie Rice, Joe Luna, who's a lead co-author on this paper with me, and we've been working closely together for the last year and a half. The Rice lab, especially the COVID team, our collaborators in the T lab and other collaborators, NIH and RU Biosafety Committee for giving us approval for BSL-2 work and the funding sources, and thank you very much.
Thank you very much, Ina, for this great talk. We already have one question in the Q&A. All of the attendees are more than welcome to write your question in the Q&A. If the speakers won't have time to answer them directly, they will be answering them later on using the text option. And the panelists, of course, you're welcome to ask questions directly. And I will start with the first question from Lucia Lifshitz. She's asking if you have tried a mix of lithium acetate with single-stranded DNA for transfection in yeast. She said that she used it to transfect yeast with big plasmids in the past.
Yeah, so actually we haven't developed the yeast protocol. That was developed by our collaborators. And this is actually a DMSO-based protocol, and it works really, really, really well and very robustly. We haven't tried anything else.
Thank you. Any of the panelists have a question that would like to ask? If now that we have more questions from the Q&A, Noam Priwas, I'm sorry if I'm not pronouncing it correctly. How does this system compare to equivalent ones for other viruses?
So I guess quite well. I don't know exactly because every virus is different, so you can't really compare them directly. It's a little more challenging to make them because of the size, but other than that, they work pretty similarly.
Great, thank you very much.